Hello everyone, it's John here. Now we're about to start a life sciences show, but we've got some very sad news. I know all of you grade 12s are probably looking forward to a great lesson, and we're going to have a great lesson, but at the same time we've got to tell you that unfortunately one of our life sciences presenters, along with his family, were killed in a car accident yesterday. So I just want to remind you, Aslam Khan was a great science teacher, a great friend of ours at Mindset, and we're really sorry that he can't be with us this evening. But still, what are we going to do about it? We're obviously going to extend our condolences and sympathy to friends and family that knew him, and we're also in that stage of can't believe it. But guys out there, grade 12s, let's make this year Aslam's year. Work as hard as you can to do the very best you can in life science to honor the legacy of everything that Aslam stood for. And so I leave you with this little introduction and hope that you will respond and send messages of support to his family. We're going to be posting stuff on the Facebook page, and we really want to wish you as grade 12s all the best, but also think about his family at this time. I'm now going to hand over to Abram, who's going to take us through some initial ideas and things that you need to know about for life science, and then we're going to get straight into the lesson. So let's all join together and remember Aslam, and let's work as we can to honor his memory. Thank you, Abram.
Thank you so much, John. It's a great loss to all of us Mindsetters and to the family at large. And we just want to send our condolences to the family. And indeed, guys, if Aslam was here, he would want us to still learn more and learn extra, guys. And I'm so happy that you guys are here to support us as well and support his family. But other than that, guys, we need to learn more and learn extra. And Cheryl is here, guys. We're here to help you guys. So let's make this an Aslam year. That's what John just said. Guys, this is the year to make Aslam proud. So guys, send your messages. We're here on facebook.com/learnextra. And also do send your questions if you'd like us to help you, because we're here to help you guys learn more and learn extra. We've got this awesome learner workbook as well that has been created, guys, to help you. More details will be coming on learn.mindset.co.za. Otherwise, guys, let learning begin, because Cheryl is here to help us. Cheryl, how are you doing?
I would like to say I'm well, but very sad today. It's a sad day.
Indeed.
Indeed it is, yes.
One thing I need to say is that we should cherish each and every moment that we get,
yeah,
and always
definitely
appreciate people. And that's one thing that Mindsetters always do with all our teachers. They always tell them how much they love them. Guys, we need to cherish, because we don't know what's going to happen next in our lives, and live a grateful life.
Definitely.
What are we doing today?
Welcome, Mindsetters. It is a really sad day today, and our hearts are really sore now. Our feelings go out to the family, but we're here to teach you, and unfortunately life is going to go on, and you've got to work, and this is your year matrix. This is where you've got to put everything in it, and that's what we're going to do today. We're starting off, all right, with the DNA and protein synthesis. I love this section, all right, one of the nicest sections. I love the grade 12 syllabus, full stop. So are we going to start?
Nice one. If you enjoy it, I enjoy it too.
Me and the board.
Nice.
All right. Okay, guys, what I'm going to do today is I'm going to just give you a quick recap of the things that you need to pay particular attention to, right? So I'm going to give you a little bit on the theory. We're not going to go too in depth, all right? And then we're going to look at the different kinds of questions that we can answer on, all right, on this different topic. Okay, we're starting off, all right, now I've got to get used to the smartboard again. Me and the smartboard are not always the best of friends. Okay, all right. When it comes to the DNA, immediately, whenever it comes to life science, you have to have this picture, all right, in your head. And when we talk about nucleic acids are rather what we call large structures, all right, that we are going to find inside a cell, right? We're going to find two types of nucleic acids. DNA, deoxyribose nucleic acid, and RNA, ribose nucleic acid. And you need to be able to tell the difference between the two. How are they the same? How are they different? And we're going to look at the role they're going to play in this process called protein synthesis, all right? An essential part of what your body does to ensure that you have these life-giving proteins, so to speak. Okay, so now let's start. Me and the pen, my very first choice is always which color I'm going to choose, and you know, people know I don't enjoy. All right, yellow, so I'm going to start pink. When we look at the structure of the DNA over here, you will always recognize it as this twisted feature. The first word I always want you to remember is DNA is what we call in a helix shaped. All right, what does helix mean? Helix means that it is twisted. All right, if you actually go back to the structure, remember it's like a whole like ball of wool. It's all twisted up and tangled, and we slowly unroll it, all right, to become the characteristic DNA shape, which then changes to become the characteristic chromosome shaped. All right, so we're going to start off with our DNA, deoxyribose nucleic acid. When we look at it, this structure is referred to as a double helix. And what do we mean by double helix? Let's have a look at a very simple diagram on the side here. What we mean is we have 1, 2 strands. That is what double means. There's a side, so when we talk about it, we always talk about it looks like a ladder. All right, that's exactly what it is. So we have these, all right, downright parts of the right strand of our DNA, and we've got these cross stripes going through. Okay, so but now we're looking at the DNA. Each of those different, my downward, all right, parts, and then my crossward parts are all made up of very specific things. Okay, let's have a look at them. Right, what do I have? I have got these little molecules, all right, on the side. That's one. I'm going to change the color. Now, these molecules on the side, all right, let's start off with, let's say DNA, all right? And when we talk about DNA, deoxyribose, what on earth does that mean? That just tells me that it's the type of sugar that I am going to find there. Okay, now this diagram, I want to use 3 different colors. 1, 2, and let's use the dreaded yellow. All right. Each of those colors now is going to stand for a particular part. All right, so let's start here. How do I know, if I look here, this looks a little bit confusing for me. Where do I know the biggest problem I find, all right, is deciding which one is the pink one and which one is the blue one. This one over here is the phosphate. All right, and this blue one over here, that is my sugar. All right, now, how do I remember which is which? Because this diagram is actually quite confusing, right? There's this one on the side. This is how I remember it. You might remember it differently. This is how I remember it, okay? These over here are what we call your, okay, these are your nitrogenous bases. So nitrogen. So we've got a phosphate, a sugar, and a nitrogenous base. How do I know which one is which? The nitrogenous bases are always the ladders over here. But this, the sugar and the phosphate, how do I know which is which? This is how I remember it. Okay? My sugar, if I eat sugar, what does it do? It always sticks to my hips. If I eat sugar, it always sticks to my hips. But my phosphate, all right, is always alone. Okay, now it starts with a P, all right. So it might sound a bit crude, when you go to the toilet, all right. Do you go in a group? No, you go by yourself. You are always alone. Where is P? P is always alone, all right. So P is by himself, but where does sugar go? Sugar always attaches, all right? So your sugar is always attached to one of the bases, all right, that goes out there. So there we've got the sugar. Now, if we have a look at these nitrogenous bases, you will notice there are these ones, or let me go to the yellow. One looks like 2, one looks like 3, all right? And those, the bases are divided into 2 groups, all right? Purines and pyrimidines. And what does that mean? Simple. One is one, all right? Here we go. If you have a look at your nitrogenous bases, your purines are 1 and your pyrimidines are 2 stuck together. Right, so that is the simple thing as my DNA. What do I have? Sugar, phosphate, sugar, phosphate, sugar, phosphate, nitrogen, 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 nitrogen. Right, and I've got 2 strands. What am I going to find here? Let's choose our last color. Joining my 2 strands in the middle, all right, I have what we've got. Let me move up and you can see we have these what we call weak hydrogen bonds, all right? Weak hydrogen bonds. Now, very often, what we mean is, the science kids say, but hydrogen bonds are not always weak. What we're trying to say when we say weak hydrogen bonds is that it is easy for an enzyme to break that bond, all right? Because what do we want to have? We want the DNA to be able to split into 2. Okay. Now, my little, as I say, I've got a son who absolutely loves Lego. All right, so where am I on the page here? Come here. Okay, there we go. My son loves Lego. And each one of these, all right, a yellow with a pink with a blue, a nitrogenous base, a phosphate, and a sugar, that is one block, all right? And that block we refer to as a nucleotide. So let's make it a bit easier here. There we go. What do we have? One nucleotide. All right? One nucleotide. Those are the building blocks. So if I were to put all your Lego out and I were to make DNA, one block is made up of a phosphate, a sugar, and a base. Block. What do I put next to it? Block. Block, 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 block. And there I've got my double strand. Okay. Now, when it comes to these blocks, these nitrogenous bases, they are very, very specific as to who they bond with. Okay? You have got 4 different types of nitrogenous bases. Okay, oh, there we go. You have got A, C, G, all right, and a T. And what does that mean? All right, they are called adenine. They are called cytosine. Sorry, I see I don't write as neat as the ladies before me. They wrote so nice and neatly. We don't write as mooi here. All right, and this is guanine. Right, we've got 4 bases. And those 4 bases, all right, make up our DNA code. When you go to the shop and you get some food, all right, and they go to the counter and they swipe the barcode, your barcode is numbers 1 to 0. 1, 2, 3, 4, 5, 6, 7, 8, 9, 0. But if they rearrange the different numbers, then you're going to have it's a code for a different price. So I use the same numbers over and over again, but if I put them in a different order, I'm going to code for something different. That's exactly what's going to happen here. All right? These are the 4 bases that nearly all living organisms have. Adenine, guanine, cytosine, and thymine. And how we put them in the order, that is how we get our different individuals. Okay, now when it comes to DNA, we have marriages here. All right? Adenine and thymine always go together, and cytosine and guanine in DNA. All right? In DNA, they are always going to go together. Now, we have got a little brother. DNA is going to make this little thing called RNA. Okay, now when we have a look at RNA, you will see, let me draw an RNA for you. All right, sugar, phosphate, sugar, phosphate, sugar, phosphate. All right, how many strands have I drawn? That's the first one. It's one single strand, it's not double. And it's also smaller, all right? So already it's different to my DNA. Second of all, I call it RNA. My sugar is a ribose sugar, not a deoxyribose sugar. So I have a different sugar. All right. My bases, A, adenine, cytosine, guanine, and uracil. What am I going to find? Uracil is a different base that I'm only going to find on my RNA. Okay, so that is how it's going to be different. Now, what do we need to concentrate on? Oops, here we go, the wrong way. Okay, this is a diagram that you need to become very, very familiar with. You can also write an essay on this, so you need to be able to tell the story from the beginning to the end. Now, as I say to you, DNA codes for proteins. What do we mean by that? Hemoglobin, pick up oxygen. If I don't make it, I can't breathe. All right? What happens? Codes for blood clotting enzymes. What happens? What happens if I can't make insulin? I'm diabetic. All right? And these things can all be life-threatening. If my blood doesn't clot, if I can't bring in oxygen, right? All of these things are life-threatening. So if I can't make them, all right, if I don't make them properly, there's going to be problems. Okay, so how do I know which protein to make? My DNA is going to tell me. My DNA is the code master. All right, so let's quickly go through the two things that we need to know, the two terms. Transcription, all right, and my second term, translation. Let's start off with transcription first. What does transcription mean? Transcription means to make, oopsie daisy, sorry, to make an RNA from a DNA. Okay, when you were in grade 10, you looked at the nucleus, and we said there's this nucleus, and it had holes in, had nuclear pores in. And we said, why is it that it needs to have these holes in the nucleus? And this is the reason why. The DNA needs to send a message out of the nucleus into the cytoplasm. And how's it going to move? How's that message going to be sent? It's going to be sent, all right, out of the cytoplasm via those nuclear pores. So what does my DNA do? My DNA, first of all, it unzips. Remember, I have, all right, it is zipped, it's double. When I need to, all right, make an RNA, what it does is some parts of it, only a small part, unzips. And when it unzips, what do we have over here? See, there's my DNA, all right? It unzips. And one of my strands has got the code, all right? Now, if you remember, this is the one thing about all these nucleotides. They never like being alone. As soon as you open the bonds, they have to quickly, all right? They need to find their partner. They are finding their partner all of the time, all right? So as soon as it unzips, we need to find the partner, and that is what we do, all right? So in transcription, what does my DNA do? It unwinds slowly, it unzips, the hydrogen bonds unzip, and what do I have? I have these, all right, these DNA template, a template, you know, like when you want to draw something that you can't draw, so you cut it out, so you use it, all right, to make it a DNA template. And what happens is, in the nucleate, there's my Lego. What did I say to you? As soon as I open up, do they like being alone? No. What happens? Complementary bases, chunk, 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 all right, they join together. Can you see here? We've got all my bases open, and what happens? The RNA bases, they align next to it. And now I need to send a message, all right? So what happens is that RNA moves out of the nucleus, and it carries a message. Because now what do I give it? I give it the name mRNA, messenger RNA. How do I know it's an RNA? You see over here, there's a U. As soon as you see uracil, all right, now, here we go. For RNA, this is how it goes. On my DNA, I've got A, C, G, T. On my RNA, A goes with U. C always goes with G. G always goes with T. With G, and T always goes with A. There's the difference. whenever I have an A, all right, whenever I have an A, I always bond it with the uracil on the mRNA. Okay, so what I've done, I made a little message. What does the message do? It goes out the nucleus, and where does it go? Okay, remember grade 10, it goes and sits on a ribosome. All right, now it's waiting. Now it's waiting. Now, when it's waiting, have a look over here. All right, is it by itself? It's by itself. Can't be, it's lonely, all right? It's lonely. Once again, it needs, all right, its partners to come and join. But if we look at it, all right, we notice that when we look at the mRNA, we can actually see it's in groups of 3, all right? And we call those codons, all right, or triplets. We prefer codons, all right? Now what's going to happen is they're alone. They're alone. They can't be alone. All right. So what has to come in? A taxi. A taxi needs to bring the passengers. All right. So inside the cell, there's a taxi. All right. And we call that taxi tRNA. You're not allowed to say taxi, right? You've got to say transfer. But don't taxis do that? Don't they carry some people and then they deliver it, yeah? It's exactly what happens, all right? Here comes my taxi, my tRNA, okay? Here's my tRNA. And what's it carrying? It's carrying its complementary bases, all right? Now, how I try to explain it is as follows. The tRNA is carrying, all right, here, you see on my head, all right? I'm carrying an amino acid. All right, so what do is, here I come. Now I've got my 3 bases because I need to know specific order. I go in, I lock, and there's my amino acid. Right, the next one comes in, it locks, there's my amino acid. Next one comes in, it locks, there's my amino acid. Now I've got all these amino acids here. Now, as South Africans, we like to braai. Here I take my kebab, all right, my sasati, and I stick that stick all the way through my amino acids. So I've got an amino acid, amino acid, amino acid, amino acid, and what do? I join them, because that's what I need to bring. I join, and as soon as I've got 50 or more amino acids, what have I got? I've got a protein. So what have I made? A protein. Ta-da!
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 You know what? After those exercises, I think you need a little break. And so to the minds, it is.
Yeah.
Well, guys, Winston Churchill once wrote that we make a living by what we get. However, we make a life by what we give. So always give. As Aslam gave so much, he was a man that was who gave abundantly of his time and teaching us as the nation and helping everybody else. So with that being said, guys, do stay tuned because after the break, learning does still continue. Welcome back, awesome guys. Now if you've got some questions, Cheryl is up and ready to help you, and so am I, guys, to relay the questions. All you need to do is to go to our Facebook page, facebook.com/LearnExtra, or follow us on our Twitter, which is @LearnExtra. Guys, we'd love to hear from you guys. What do you think also of our brand new set? It is for the first time that we're here, Cheryl.
You guys see a very different picture than you do. We're in a virtual set, okay? Now, for those of you who are a bit as doff as me, everything's green. There's nothing on the wall, but you are seeing a new picture. So it's a virtual set. So and it looks absolutely wow. So if you comment on our whole new setup here, so as I said, those of you who boffins, you know exactly what I'm talking about. Those of you who don't worry about it. Just as long as it looks
It's
cool.
a pity we are live. We can't see ourselves.
No.
We're seeing a different picture. Well, for people like Cheryl, the BTs before technology, well, guys, we just need to say it's just beautiful. But for us, 80s after technology, we're loving this new look. I know that you love it too. So if you give it a thumbs up, let me know on our Facebook page, just write on our wall and let me know what you guys think. But still on a happy mood, we're giving away a car, guys.
Oh,
on the Beer Bright Spark competition. Already we gave 1 car away to Dimakadzo last term, so you could be next. I could be visiting you and say, hi, Cheryl. Unfortunately, you're not
Yeah, I
allowed
know,
to enter.
I
You can
know.
say hi, and yeah, congratulations, you've won yourself a Chevy Spark L, a beautiful car. I'd love to win one, but I just can't. But you can, guys. So all you need to do is to download the Cool Android app, which is available from Samsung Galaxy Store or the Google Play Store. It's cheap, guys. It's affordable, guys. 59 Rand. You can get it right now. Otherwise, let's continue, Cheryl, because I
Hey,
can talk forever.
And I can see that. You just called me a BT. Just you watch afterwards.
no.
Just watch the space, yes.
I take that back.
No, I am a bit, yeah. Okay, guys, now we're going to look at the different kinds of questions that you can see, all right, or you need to be able to experience when we're doing the DNA. I won't say I'm going to go through them quite quickly, but I just want to show you the kind of questions that we can ask, all right. The first question relates to the actual structure of the DNA. All right, so let's go through our question. All right, it says the diagram below represents a part of a molecule. Now, it doesn't tell you that because sometimes it wants you to see, is it a DNA or is it or an RNA? And then you need to give reasons why. All right, now it says here, study the diagram and answer the questions that follow. All right, if you have a look here on your very first thing, most of you, all right, what you should looking at, we can see, okay? I think it's a DNA molecule. Why? Because I can see that it's got 2 strands, all right? But I'm not too sure. I can't see, it doesn't give me, all right, all the different bases totally. I've got adenine and I've got guanine. So I am certain of it, but let's go and answer the questions first. All right, the first question was identify the molecule in the diagram. So as it says here, it doesn't say anything about uracil. And as you can see, all right, A goes, this will be T, C there. So then this will be A, that will be T. And if you have a look at the shapes, you will see, all right, that it's adenine, cytosine. Also, as I said, it's going to be double-stranded. Okay. All right, so if I were to identify, there we go. For 1 mark, nice and easy, I would say it's a DNA. The second question asks to label the parts numbered 1 through to 5. Okay, so as I said to you, all right, I need to keep on chopping and changing. Number 1, well, I'm going to mark it on here, is my weak hydrogen bond. All right, number 2, it's by itself. If it's alone, we know it's a P, all right? So it's my phosphate, which then makes number 3. Now, because it's a DNA, I don't want to be too, but I would prefer it if you were to tell me what kind of sugar, all right? It's a deoxyribose sugar. Okay, be specific because it's a DNA. My number 4, I can see what always goes with adenine, all right, is my thymine. And number 5, what always goes with cytosine is my guanine. All right, so there we go. From that, it's a nice simple diagram. Are you able to label the different parts of the DNA? Okay, we go back to my arrow. There we go, nice little open box. There we go, my arrow. Oh, okay, it's not opening. There we go. All right, so numbers 1 and 5 respectively, if I go back up, 1 was my hydrogen and number 5 was my guanine. Okay, now it says, what is the collective? All right, collective, sorry, I got to keep on jumping from the one to the other. I apologize. To be able to open the blocks, I can't write and open the box at the same time. Multitasking. Right, what is the collective name? All right, for the parts number 2, 3, and 4. Now, as I said to you, we have a group. 2, 3, and 4 is a base. What is that building unit called? It is a nucleotide. All right, a nucleotide. Many nucleotides, those are my Lego blocks, right? Those are my building blocks. They are going to, all right, make up, there we go, a nucleotide. Question 1.4. What is the significance of this molecule being able to replicate itself? All right, now this is a very, I'm going to put here, let me draw again, okay? I'm going to say it's an important question. All right, and I'm going to go back the answers. Why is it essential? Right? Here we go. When I talk about a replicate means to make an exact copy. Why do I need to make an exact copy? Well, let's think about it. What we're going to be doing, all right, what we're going to look at later when we do your genetics and all of those things is a process called cloning. Now, when we think of cloning, we think of all animal cloning, but no, asexual reproduction. Plants reproduce, lots of plants reproduce by making exact copies of each other. How do they do that? They need to make an exact copy of their, all right, of their DNA. What else do we need to do? We need to make sure that when, oh, come on, you're me and the smart board again, all right? We need to make sure that those hereditary characteristics, when we make a copy, can you remember mitosis, all right, when we did it in grade 10? What was mitosis? The cell made an identical copy of itself. How did it make an identical copy? It made an identical copy of its DNA. All right, what do we also need to make sure of? I'm sure you guys have been told about the Human Genome Project, all right, where we map every single, all right, code in every single trait that we have in our genes, right? That we need to pass on, that we need to know, that we need to make sure that it's correct. Because remember, later on you're going to do, if we have a change in DNA, we have a mutation, right? And a mutation, not necessarily bad, but usually it is. It can cause, all right, quite a lot of problems, all right? So when we look at our DNA, all right, there's lots of reasonings over here, all right, that you must make sure that the cell, all right, an organism, to be able to reproduce, needs to survive. If you grow, if you damage a cell, all of those cells, all right, need to be able to make identical copies of itself. Okay, let's go on to question number 2. All right. Where is DNA found? Okay, nice little simple questions. All right, let's start. Where do we find the nucleus? Aha, all right. Some of you, I don't know if you got to that section yet, will know about that some DNA is found in the mitochondria, mitochondrial DNA. You're going to learn about it a lot more, right, especially at the end of the year when you look at evolution, all right, when we look at how mitochondrial DNA, we use it as a bit of a GPS, all right, of our history. Now this is just different ways that we've asked them. What are the constituent groups of molecules that form the nucleotides? Just now I asked you, what do those three, what do they make as one? Now they're saying to you the opposite. What does a constituent, okay, sorry, let's go back here. Constituent group means, what does it consist of? All right, okay, and now my answer. All right, what are my 3 things? Yes, a phosphate, a pentose sugar, all right, nitrogenous base. When we say a pentose sugar, that means a 5-carbon sugar, all right, and that's your deoxyribose or your ribose. Now it says, name the mRNA produced by each of the following strands of DNA. All right, what on earth do they mean by that? Here is a code. What am I asking you to do here? I'm asking you to tell me what is the matching strand. What does A go with? Oh, now hope here. A goes with U. A goes with U. C always goes with G. All right, so let's carry on. Have a look there. Right there, guys. That is the most crucial thing. Whenever you get asked to make an mRNA, I never want to see a T. T stands for taboo. All right, no go on mRNA. Right, I only find T, all right, on my DNA. And that's going to cause you problems if you keep forgetting that. All right, so you need to be able to do 3 things. You need to be able to have your DNA code, all right, change it to an mRNA code, and your mRNA, you need to be able to work out your tRNA. If I give you, this all sounds like maths, I know, and maths is scary. If I give you one code, UCG, if I give you one code, you can give me the DNA and you can give me the RNA, all right? There, A, G, C. Over there, it's always the same, A, G, C. So I just need to give you one code, and you can give me all the others. Right, okay. So be able to, there we go, nice easy marks. Okay, C goes with G, there we go. A, U, A, U. I never find a T. T is taboo, all right? T is taboo. And there we go, all right. So those are the two different kinds of questions that you can ask. So one is just a general structure, right? And here, right, we should be going. You must tell me when we need to take a break.
I think we should take a
Are we
break.
going to take a break?
Yeah,
Okay.
I know, because you can just go
I
on
can just
and on.
carry on forever.
Well, guys, I
Yes.
think you deserve a break. And during this break, make sure that you send through your questions to us on our Facebook page, which is facebook.com/LearnExtra. Don't forget, guys, that on our page, it's not just about us. It's about all of us. We're a family. We should help one another. If you see someone struggling on the page, make sure that you respond to that, and let's help one another. Be a helping nation and a caring nation. Otherwise, we'll see you after this break. Welcome back, awesome guys. I hope you're still with us. If you just joined us now, where have you been? Because Shaw has been on fire. So are the other
Yeah.
mindsetters on the
Yeah.
page. So guys, if you just joined us now, welcome, welcome. Send through your questions. We're here to help you. My name is Abram, and I'm with Cheryl. Just to tell you more, guys, about this workbook that we have. It is CAPS aligned, and it was developed by Cheryl and reviewed by Aslam, guys. So this is an
Yeah.
awesome, you rest assured when you have this. That's what most of you guys have been asking for, and Mindset has heard you guys, and this is what we're doing for you guys. So more details on where you're going to get it, how you're going to buy it, are going to come through on land.mindset.co.za. How has the journey been, Cheryl, in putting this together?
As I said to you, I was looking at Llewellyn and I both did it last year. I didn't realize that it would go into copy and whatnot. It's like having a baby.
This
a lot
is
of
your
hard
baby.
work, but it's wow. I'm actually, as I said to you earlier, I'm quite taken aback. I'm proud.
It's a great gift
Yeah.
to you guys that will never perish. So
Yeah.
guys, if you're going to get hold of this, be rest assured that you're going to pass with flying colors. But for now, let's continue with the questions.
Please also, I'm skipping through because this hour is such a short one. I've skipped a few questions, all right, just so that I can give you the general trend. But if you go into our notes, all right, if you go into, I think, is it on the web and whatnot?
Yes.
I've put the notes up. All right, this is Aslam's. Aslam put the notes up. I'm obviously standing in his place, but he's got the notes up, and all these different kinds of questions are there with the answers. So guys, you've got all these resources. Make use of them. Okay. The next question that I want was only for 3 marks, but I want you to have a look at something. Right. One of the things that you did for DNA. It says here, a child is born out of wedlock. What that just basically means is the mom wasn't married, and the mom can claim maintenance for the child, but she doesn't know who the father is. Okay. Blood tests were done, but both men had the same blood type. You cannot prove paternity, all right, by blood tests, because blood tests will just give you a general group, all right. If they are the same blood type, obviously here you're going to need to do further. So what she did next, both men provided samples, and the VNTR of both is analyzed. The results are below. Okay. Now what do we mean is this? Remember I told you about barcodes. That's exactly what we're talking about. Right, how do we test DNA? We take little snippets of DNA, all right, and what we do is you all watch CSI, and at the end we put it through a whole, all right, process of gel, right, of these electrolysis, all right, and all the different segments they separate. What we're looking at is a DNA fingerprint. Please remember it's different to this fingerprint. Okay, this is your fingerprint ID. A DNA fingerprint means it's a strand of DNA showing you the codes. Now, if you have a look at this diagram, have a look at the headings. There is the mother, there is the child, there is the 2 males that were in question. All right. We're going to take the mother out of the equation now because generally, all right, we do, if the mother gave birth, she wants to find which of these 2 are going to be the dad. All right. Please make sure you take a ruler when you do that, and you take your ruler, all right, and you check to see, all right, where they are the same. So if we have a look here, the top one, imagine it's a ruler, it's a thin one, those don't match. Then we have a thick one, but this male has got 3, but this has a thick. All right, yes, we match. We've got one match. Here, do we have a match? No. Here, do we have a match? Match, 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 match. All right, do I have a match? Do I have a match? If I look at the bases, all right, it looks that I could. It's thinner, all right. Where do I see? Where do I have a thick one? Yes, there I have a thick one. I have a thin one. No, no. Sounds really weird, right? But what I'm trying to do is I'm looking to see what the child has and what the other two have. No, 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 no. But I have a yes and a yes. And if I look at it, if I have a look at it, what do I notice? All right? That male number 2 over here, has got far more of the same segments, all right, than male number 1. So because of that, all right, I can then conclude, all right, that the father is number 2. And the reason being, okay, there were 5 matches in the VTNR, right? So we must, each time you must look, right, you have a look, it's the barcode. If I've got a number 1 here and a number 2 and a number 1, one's match. If I've got, so what happens when you can't find the price at a store? Right? What happens when you can't find the price? What do? All right? What do? I look to see, okay, here's the barcode here and here's the barcode here. Do they match? So obviously, the closer they match, all right, when it comes to paternity, that is going to tell me that they must be more closely related. Okay, all right. Now, as I said to you, yes.
On that question, you have found 4 matches.
I think I should have found one more. Should I have found one more? Okay, I said 1.
Because I think I'm more interested in this part.
1, 2, 3, 4. Let's find where if I missed one match. 1, 1, 1, 1. I think this one's a thick one, thick one, thick one, thin one, thin one. No, there's only 4 matches. Okay. All right, there's only, no, if we go see the thick and the thin, they don't go. So we'd say there's 4 matches. But irrespective, okay, 4 versus 1. All right, that's how they would be able to do it. I think in South Africa, when they do the paternity test, I think we check for markers out of 10. Right, and the closer you have, I think you have to be an 8 out of 10 for it to be the matching of the DNA. All right. So obviously, where do we use this process? Right, if we can't identify the human remains, they want to use this process in rhino poaching. Right, so if they got the DNA of a rhino, they can match the horns and poaching, etc.
So they've got a certain number that they look at. If it's
it all depends on for it, or you can do longer ones as well. But you know, it all depends on your test. But obviously, the more you've seen it, and it comes like a segment because they separate. Yes. All right, so they separate like that, and the more of these bands, and the more you have in common, obviously the more closely related you are going to be.
Makes sense.
Okay, all right. I love it when you ask questions. All right. Okay, now as I said to you, this is a diagram. You need to become, all right, for me, I always take that diagram and then I tell a story about it. So I go from the diagram, what happens, put numbers to it, all right? Because as I said, you'll see the last question here, we're not going to have time for is an essay, right? In an essay, you need to be able to plot your thoughts, all right, step by step. Okay, now it says here, okay, all right, it says here, always complete the labels on the diagram before you move on to the questions. I like to do that. You know what? Sometimes they don't always ask you to, but already you've got a picture. Start to fill in. What do you think they're going to ask you? All right. So let's start over here. If we were to do, as I said, these are Aslam's notes. So he's like this voice is telling us where do we go. All right. So what do we look at? What do we need to know? We need to know process A. So here's the word DNA template. And here, let me get my pens with me again. DNA template, and here I see a single strand. What is my process? Transcription. Because what is transcription? The making of a RNA from a DNA. Okay. Now it says process B. Process B, here's my taxis, all right, my tRNA, What are they bringing in? What's going to be on top of them here? They're going to have amino acids. So what are they doing? This is my code. I now need to translate my code. All right, that's what translation means. All right, doesn't help me if I've got a code and I don't know what it means. All right, they're going to tell me what my code means. I see this is telling me an organelle here. All right, this is going to be my ribosome, because where does my mRNA come and sit? Onto a ribosome, right? And these are the amino acids. And amino acids, when I join them all together, I make this thing called a protein. All right, so let's keep me and this little board over here. Okay, so let's start. Okay, let's go back to the hide there. Number 1, name the following process. A, what did we say happens at A? Let's see if we're right. Process A, transcription. Process B, translation. What? 1, 2. Well done. So far we got 2 out of 2. Name the organelle. Remember the language. An organelle is a small little structure inside the cell, right? And my organelle involved is my ribosome. All right, there we go. Oh, so far we are doing so well. Ha. Now we get to our long questions. Explain how the mRNA is made from the DNA template during process A. They only want you, all right, to describe what process? Transcription. Okay, now the biggest problem when it comes to life science. We have to speak, oh, did I go? Come down here, you. Okay, let's go to the arrow. The biggest problem here is we need to speak biologically. All right, so you need, first of all, the process is called transcription. All right. Now, free RNA nucleotides, all right, in the nucleoplasm. They arrange themselves according, so let me go back to the pens. Okay, these are the things you need to be able to use, all right. So while DNA replication takes place, or you can say the DNA unwinds or unzips. Okay, there we go. Now, what is happening? Free nucleotides in the nucleoplasm. See the words that I'm using. That's where they come from. Where do they go? They arrange according to the base sequence. What is that telling me? They find their complementary partners. All right? A goes with U, C goes with G. What happens? The sugar and the phosphate bonds between the nucleotides, and what do they form? An mRNA. And what is this process controlled by? Enzymes. That's where you stop. How are we doing for time? Have we got a little bit more time?
We're doing well.
Okay, cool. All right, now, process A and B. Now this is where, all right, you need to be able to have a bit of extra knowledge. Process A and B can be summarized by the table below. Write the numbers 1 and 3 next to each okay, let's go over here. You'll see when we see the question. All right, the table they have, there we go. Have a look there. All right, remember what I said to you? I only need to give you one of the codes. The DNA, the mRNA, or the tRNA. And from that, you will be able to give me any of these. All right. So, CAA, all right. CAA, what goes with C? G, oh, there we go, pen. What always goes with C? G, come guys. Okay, there we go. G, see? G. What always goes with A? U. U. mRNA is always a U. Okay? And what is the tRNA? The tRNA is always the same as DNA except with the U. So what do we have? CAA. All right. Now work backwards. GCA. What does G always go with? C. What does C always go with? G. Now it's on the DNA. what does A go with? T. All right, T. You always need to be able, if I give you the one, to be able to work backwards or to be able to tell me what the other one is going to be. Okay, I want to show you just one more question on the DNA. Okay, here again, protein synthesis. Can you see? DNA is unwinding. All right, can you see that? What do I make? All right, I'm making an mRNA. Let's go what Aslam said to us. Let's start to label. All right, what are those little groups called? Codons. All right, now if that's a codon on the mRNA, remember, that's called an anti-codon. Oh, can you see it's what's flying in? What is flying in? My taxis. Right, remember not to say it, but think it. All right, okay. And what do the taxis carry? They carry an amino acid. Where does this process take place? On the ribosome. What's this process called? Translation. What is this process called? This is called transcription. Where does it occur? In my nucleus. There's my different organelles. Okay. All right. So if I were to provide labels, okay, there we go. N is mRNA. I think we labeled it. There we go. mRNA. P was my amino acid. Q, all right, over there, all right, was my tRNA. Sorry, I labeled on that one. Okay, and then lastly, my Z is a codon. There we go, group of 3. All right, write down the sequence of the first 3 nitrogenous bases on the DNA strand that led to the formation of Z. Now look here, look. Here's Z, look at this, I'm going to use a different color. All right, here's Z. Z is this, UCA. All right, they want you, what was it on the DNA? What is U? U always goes with A, C always goes with G, and A always goes with T. Remember, this is the mRNA, this is my DNA. Always, it is so vital. It is so vital for you to remember that little thing. On the DNA, I have a T. On the RNA, T is taboo. All right? Taboo means no go. Okay. All right, so there we've got our sequences. All right. Name the part of protein stature that is illustrated by O. What did we go up? The O over there. All right, what is it? That is when the DNA unwinds. All right, and we call that process transcription. How are we doing for time? Oh, okay.
I think we're almost done.
We're almost done. Because I want to show one quickly. All right, guys, one more thing quickly before we go. All right, you need to be able to know the basis. All right, because what you can do is they can ask you the different, you never have to know the amino acids. All right, but if you can work out the different bases, you can then work out the different amino acid. Now remember, amino acid, all right, let's go pen. Where do I find the amino acid? On my taxi. See, there's my taxi. All right, there's my taxi. All right, and what is my taxi? Sorry, tRNA. All right, and here is my DNA. So if I've got my DNA, I can work out what my RNA is.
Right.
We could have carried on.
That's all we have
time
for. But thank you very much,
thank you, Abram.
for us. I'm sure. Thanks to you guys for tuning in, and thank you for the messages that you guys have been sending on our Facebook page. We really appreciate them, guys. Thank you for your thoughts, and thank you also to our sponsor, which is MTN SA Foundation, for proudly sponsoring this show and bringing it to you guys, and of course the Department of Basic Education for making sure that they support this wonderful show, guys. It doesn't end there. If you need some help, we're here for you guys. Go to our Facebook Facebook page, facebook.com/learnextra. We love you and we'll always be with you guys to help you and to carry you through, guys. Remember that you are the best that you could ever be. Be yourself. We love you. Peace.